recognizing the critical role of science and technology in economic development and progress. The Balik Scientist Act, or Republic Act 11035, was signed into law last June 2018 by President Rodrigo Roa Duterte.
This is actually putting into law a program that has been started by the Department of Science and Technology almost 40 years ago. But we need some legal support so that we can implement it in a better way. The Balik scientists will have an easier time in terms of coming here to the Philippines and rendering services.
Who is the Balik scientist?
Experience and expertise of Filipinos who have made good practice of being scientists abroad, so that they can share whatever they have in terms of knowledge and wisdom to our own institutions, our own researchers.
My role and responsibility of a Balik scientist is to be not just a teacher or facilitator, but also a pusher of innovations within the program.
Balik scientists are given support by the government for their stay in the country and are likewise provided with a wide array of benefits to ensure their maximum output.
The best incentives or privileges or benefits are having to be exposed with our farmers.
My greatest privilege would be doing collaborative work with fellow Filipinos.
The DOST, as the leading agency in charge of the Balik Scientist Program, is tasked to facilitate the placement of the Balik Scientist among its priority areas from its sub-agencies. P-Card, PCHRD, P-Shared, Partnering with the DOST are the host institutions, private or public entities, providing the appropriate resources to the Balik scientist in the completion of their research activities and other tasks.
I think the role of the institution is to give the space or the laboratory needed for the program or the project.
Working together, the DOST, the Balik scientists, and the host institutions have proven the importance of collaboration and cooperation, critical of any nation's vision for success.
I am an advocate of Balik Scientist Program. Okay, being one, I really truly felt that Balik Scientists would be able to help in uplifting the economic growth of the country.
My hopes is to really contribute to the space agency. It's very, very pragmatic, and we will need everybody's help, and also promulgate the STEM program here in the Philippines.
We really need to encourage other Balik scientists or other scientists abroad to give their time. They need to give back and help the country.
With the enactment of the Balik Scientist Act, the country is looking towards a stronger and more solid science and technology foundation, propelling the nation to further heights. Change has come indeed for science and technology. Science for change. Science for the people.
Okay, so I'm really sorry for the technical problem earlier. Let us again check our number of participants. Here in Zoom, we already have 91 participants. And in Facebook, we have 31 participants and 5 in YouTube. Okay, so to formally welcome our distinguished speaker and our dear participants, may I please call on Dr. Annabel B. Briones, the Director of the Industrial and Technology Development Institute, or ITD. Dr. Briones.
Thank you, Ms. Athena. Am I loud and clear?
Yes, ma'am. Yes.
Okay.
Loud and clear.
So our distinguished speaker, Balik Scientist, Dr. Leodivico L. Ilag, my ITDI family, VIP ITDI team, DOST officials, colleagues, participants, guests, my warmest greetings to all of you. I want to welcome all of you here today and thank each of you for participating in this activity. Today is the 7th webinar of the project on establishment of the Virology and Vaccine Institute of the Philippines, or VIP in short. We are fortunate to have 7 Balik scientists who will share their expertise in virology. This webinar series will run next year. The DOST VIP program was conceptualized to address the pandemic readiness of the country. The ongoing COVID-19 pandemic presents many concerns, particularly effective clinical and public health management, primarily on novel viruses. So these issues can only be addressed using science and technology, specifically through research and development. Thus, DOST finds a solution to this global concern by establishing a Virology and Vaccine Research Institute with the primary goal of developing diagnostics, vaccines, and therapeutics. In addition, the Virology Institute will be the venue for scientists, both here and abroad, to work collaboratively to study viruses. The objectives of the VIP is to serve as the premier research and development institute in the field of virology, encompassing all areas in viruses and viral diseases in humans, plants, and animals. Second is act as a venue for scientists, both here and abroad, to work collaboratively to study viruses of agricultural, industrial, clinical, and environmental importance. And the third is to establish strategic partnerships with the world's leading scientists, virology centers, and institutes, and conduct innovative and pioneering research that will advance the frontiers of the virology in the country. To institutionalize the VIP, we get the support from our lawmakers, both in Congress and Senate, by passing the bill regarding the establishment of VIP. And while we are still waiting for the bill to be enacted into law, the DOST has initiated several R&D projects implemented by ITDI, St. Luke's Medical Center, and the Research Institute for Tropical Medicine. So these R&D projects are being implemented in partnerships with several local and international researchers and institutions. With these initial projects, we hope to build the capacity of the VIP and help resolve several of the pressing issues in the country brought about by viruses. And part of the project's activities is engaging the expertise of the Balik scientists to help us accomplish these initiatives. Dr. Leodivico L. Ilag will discuss the early interventions against COVID-19. With that, I thank Dr. Ilag for accepting the DSP engagement and helping ITDI in the VIP program. My special thanks to the VIP team of ITDI, the Technological Services Division, and the Planning and Management Information System Division of ITDI for organizing this event, the BSP Secretariat, or the Balik Scientist Programs Secretariat of the PCHRD, and Picard, our ever-supportive Secretary of the DOST, Fortunato T. de la Peña, Undersecretary Dr. Wena Cristina L. Guevara, Director Jimmy Montoya, and Director Rinaldo Iborra. I encourage the participants to join the Q&A portion actively. I hope you will learn a lot from this webinar. Again, welcome and a pleasant afternoon to all of you. Thank you very much.
Thank you, Dr. Briones, for the warm welcome for our esteemed guest speaker and our participants. So now let me please formally introduce our distinguished Balik scientist and resource speaker for this afternoon. Dr. Ludovico or Vic L. Ilag obtained his bachelor's degree in biology, magna cum laude, at the University of the Philippines, Los Baños. He then pursued his PhD in microbiology and immunology at the University of Tennessee Center for the Health Sciences in the United States of America. Thereafter, he served as a postdoctoral research fellow at the Purdue University in the USA, working on the structural biology of viruses and viral proteins. Dr. Ilag has more than 25 years of solid research in the discovery, preclinical, and clinical development of biologics and dietary or food ingredients such as peptides, proteins, antibodies, natural products, and agricultural waste streams. With applications in oncology, cardiovascular disease, CNS, infectious diseases, metabolic diseases, and dermatology, and technical experience in antibody engineering, structural biology, virology, immunology, biochemistry, microbiology, and genetics. Being a virologist for more than 2 decades, working on institutions here and abroad, Dr. Ilag has more than 30 publications on virology and immunology and in peer-reviewed international journals, several presentations in national and international conferences, and several patents under his name. He has worked on the characterization of antigen-antibody interactions, different pathogens, and how these may be utilized in the development of diagnostics and therapeutics. An expertise that we earnestly sought for in the implementation of the initial research projects of the VIP. In his long tenure as an academic, Dr. Ilag has worked on several projects characterizing the mechanisms of viral infections. Aside from Dr. Ilag's experience as a researcher and an academic, his experience as a member of the top management and several international private institutions allowed him to see the needs in the ground in terms of diagnostics, therapeutics, and vaccines, either for medical or veterinary purposes. During his tenure in the private industry, he was able to successfully lead the development of a functional proteomics platforms involving immunization, generation of antibody fudge library, selection of novel antibodies, and the like. Ladies and gentlemen, let us give a warm welcome and a big virtual round of applause to Dr. Vic Ilag. Dr. Ilag.
Okay, thank you, Athena, for the kind introduction. Let me just share my screen. Yep, so since I'm actually broadcasting from Melbourne, there's a tradition that I'd like to show respect that I'm on the lands of the Wurundjeri people, and I wish to acknowledge them as traditional owners. I'd also like to pay my respects to the Wurundjeri elders, past and present, and Aboriginal elders of the other communities who may be here today. All right, so going to the slides. So as Annabelle described, as well as Athena, I'll be talking about the implications of early intervention of SARS-CoV-2 virus with implications to the disease itself, as well as the variants. As full disclosure, I'd like to make sure that I'm not offering medical advice in this talk. I'm not a medical doctor. It's mainly purely looking at the scientific basis of infection as well as the disease. So SARS-CoV-2 virus is the etiological agent which causes the COVID-19 infection. I'm not going to go through much detail, but some points I would like to highlight later. We'll discuss about the eclipse period, burst size, and some things to highlight that this virus stays in aerosols for about an hour. The surface is about 1 to 10 hours, and the antibody response takes about 10 to 20 days. So the way I structured this talk is the first part will discuss more the biology of the virus, its mechanism of how it infects, and then move to the second phase on the early intervention strategies and the implications of why it's important to intervene early in this viral infection. So like any other virus, SARS-CoV-2 essentially performs similar basic functions. There would be variations in a theme because viruses infect different hosts, but essentially you could break them down into three steps. The virus, because it's an obligate parasite, that means it requires a host to continuously multiply. So just like any biological system, it needs to propagate itself and continue its legacy by continuously replicating or reproducing, and so that it could multiply continuously. So this could be broken down to three steps. So the virus has to bind and enter the cell. And once inside the cell, it uses the host cell machinery to produce more viral particles. It has to replicate. And once you have a lot of viral particles, these are then have to be released. So there's essentially a control mechanism wherein the virus has to take charge of the host. And one could essentially divide this process as a first part where you have the binding and entry of the cell, as well as entry into the cell and multiplication of viruses and host control, determining the destiny of how the host could coexist, or how the virus could coexist with the host and to produce more viral particles. So from a clinical perspective, so once one is infected with SARS-CoV-2, essentially people have called this a biphasic disease because essentially there are two phases. The first phase is once the virus infects, you have a host virus response. Essentially, this is the time when the virus multiplies, replicates, and produces most of its more progenies or more viral particles, so that it could infect other hosts. It then transitions as the time passes by, usually 7 to 10 days. You have this interface where then the host develops a defense mechanism, and usually it's through that host inflammatory response. And at this stage, you have like a point of no return, what I call the Rubicon, where then at this stage, it doesn't involve any viral replication anymore, but just what you call, and often people call this the cytokine storm. And this is the reason where most people die from it. So this is the severe phase. So just from this slide, one could already tell that the importance of inhibiting or controlling viral replication early on. So of course, you can see from here that antivirals are the major mechanism of how to prevent this phase. And of course, the major effort as well as we see is the development of vaccines. So obviously, from this slide, one could see that if you intervene early, you prevent the severe phase from happening, therefore you prevent death. And for this virus, it's known that 99.97% of people who get infected survive it. They've had mild symptoms, but for those who don't make it or enter the severe phase, then and those with comorbidities usually develop this host inflammatory response and cytokine storm, and usually leads to death. So I'm going to tackle these two. So that's two phases. And let's now move to the molecular basis or how this works. So without going into too much detail, just go view. So we have the SARS-CoV-2. It binds the receptor. There are two of them, main receptors, the ACE2 receptor. There's a co-receptor, TMPRSS2. And then once it gets clipped to allow the invagination or endocytosis of the virus into the cell so that it could enter it. And once it's inside the cell, it gets released. It releases this RNA molecule, which encodes the information to produce a protein, a long protein. And the first thing happens is the protein clips itself, a portion of it, a protease, which chops it into small pieces, which correspond to different viral proteins, which have different functions. Some of those proteins enter the control cell or the control command center of the cell, which is the nucleus, to prevent or delay the immune response, the defense mechanism of the host. At the same time, some of the proteins then are involved in replication. Essentially, what happens then is that the virus now takes control of the cell, and the primary objective is to manufacture and usurp all the components in the cell, amino acid, nucleotide, to produce more viral particles. So from the time the virus binds to the production of the first infectious particles, what you call the eclipse period, and that takes 10 hours. So as the virus continuously replicates and produces more viral particles, the cell starts to get sick, and then it tends to send signals, what you call cytokines, and this is where you generate the cytokine storm. So in principle, that's just the overview of how the virus replicates. Okay. So now let's shift a bit the implications of viral replication and what it means. So if you, let's say, just for the example here with SARS-CoV-2, we know that within 12 hours, with an eclipse period of 10 hours, roughly 1,000 viral particles or virions, infectious viral particles are produced. So let's say if we assume that only 10% of these newly released viral particles will then infect neighboring cells, you will generate about 100,000 virions. So after 2 days, you would roughly have 1 billion virions or infectious viral particles. Now, if we assume that only 1% infect of the 1,000 virions, in 2 days you have about a 10 million virion. So this is just like to highlight here. This shows you that from one viral particle, you could produce 10 million to 1 billion viral particles in 2 days. Usually symptoms appear around day 3 or day 5 by the time you're infected, but you're already starting to shed viral particles as shown in this slide. What's interesting about this virus is that actually it has a mutation rate. So it has developed a mechanism of making errors, not for the sake of making errors which are detrimental, but it's a means of actual survival of the species, of the viral species. So if we assume that the viral genome of COVID-19 or SARS-CoV-2 virus are 30,000 nucleotides, if the virus is making an error every 1 millionth of a base or nucleotide, every 34th newly made virus will be a variant or will have a mutation. So if we translate that here, you would see that from this, you'll have about 30 million or 300,000. So roughly just about under 3%. So from a population, after 2 days, you're already generating variants. So if you continue this for 5 days, you could imagine then that you'll have billions of variants, billions of viral particles, and about roughly 1% of that will be variants of the composed of variants. Now, one of the things I'd like to point out here, that most of those variants would have mutations which are deleterious, which means it will lead to a non-functional virus. So that won't have any effect. Some of them would have mutations which doesn't change anything with the virus because of the degeneracy of the genetic code. Therefore, they just behave as the wild type virus or the original virion here. However, there would be a small fraction of viruses which would have mutations which would still be tolerated by the virus, but gives them selective advantage. That means it could either evade the host or what you call, and I'll discuss that later, mutations that evade other mechanisms or even stabilize the virus, or most likely one of the early mutations of virus would be favored would be those which could increase its burst size. So instead of making 1,000, probably could make 1 million particles per infection. So now what's the concept of the viral resistance as well as escape? So in a natural population, I'll just show you the example. So in a natural population, you'll see here that there would be a small portion would be variant. So most likely one would be infected by the wild type or the original virus particle. However, there would be instances, there would be selective pressure. So whether it's temperature, let's say, if the virus is exposed, it then has itself be more stable in temperature, and the wild type or the original virus is less stable, then there would be a selection for this. And what will happen, there will be enrichment, and most of the viral particles which will be replicated will be the variant. Another example of an escape would be those who have antibodies, let's say only against this particular wild type virion, and for some reason the variant would have a mutation in a site where the antibody binds, and the mutation is subtle enough that it doesn't inactivate the virus, but it actually prevents now the antibody from binding it. So this is what you call escape mutants, and it's actually common with what you see with the vaccines. And with that you would then have a selection, and there will be enrichment of these variants were then escape. Now, I'd like to point out from this slide, just like to show that it's important to emphasize that selective pressure does not cause the variant. So there's this misconception, I think people are saying that if you then get immunized, let's say for the example of virus for vaccination, so antibodies itself does not cause the mutation, but it already pre-exists. Antibodies or other agents could create selective pressure so that could lead to enrichment. So that doesn't mean that vaccines are bad, that they create mutants, but they rather enrich for the mutants, which have implications which I'll show later. All right, so now I'd like to shift here a bit in comparison to dengue. So dengue is another virus. It's also an RNA virus, but it's different. It's unlike COVID-19 or SARS-CoV-2 virus, which is a respiratory virus. We know this is a bloodborne virus, and it's transmitted by mosquitoes. The reason I'd like to show this example is to demonstrate the importance of, or a strategy of preventing this from being a pandemic or spreading. And we know that we already pretty much have a good control of dengue. Once in a while, we have some kind of breakouts, but most of the time, it's already controlled. And the reason that is the case is because of the strategy which we have developed for dengue. By understanding the mechanism, we know that the virus essentially replicates within the mosquito, is transmitted by the mosquito. Therefore, the key vector here to eliminate or to control is the mosquito. So after understanding essentially what the way to manage dengue infections is, you find areas where mosquitoes breed, you destroy those areas where they're breeding places as well as mosquitoes, and you prevent the breeding of certain or the breeding or the propagation and multiplication of viruses through the mosquitoes, because that is the vector. Now let's do a bit of a thought of experiment here, where the question is, what if we use the current COVID-19 approach against dengue? So what do we know about COVID-19? We know it's airborne, mainly transmitted through aerosols. It does happen with droplets because it's heavier than your contact transmission. But recent studies shown with the CDC that the probability of getting transmission through droplet contact transmission is very, very low. And most of the transmission is happening through aerosols. And based on that, and if you compare that to the current methods, so I think once you test positive, the major efforts to control the disease is to use these different methods. We use masks and shields, social distancing, better air circulation, quarantine, and washing hands. Now, washing hands is important, especially for contact transmission, as well if somebody touches their mouth and then could contact that disease either through the nose or through the mouth. However, if you look at the disease itself, looking at dengue, you would notice that we don't have a find, destroy, and prevent strategy. Anybody who has masks and shields, in spite of having 2 or 3 masks and shields, does not stop the replication of the virus. Social distancing actually only works if people do not move, because once somebody's in point A, that person breathing the virus is forming aerosols in spite of having masks, would still be able to have the air, still have aerosols in the air containing the viral particles. But if that person moves out of position A and somebody moves to that new position, there's the chance that that person will be getting that virus. So it's not, social distancing per se is not sufficient. Now, I'd like to point out here, now I'm not saying that I'm against these strategies, but the strategies itself shows that it does not prevent or destroy the point of the vectors of transmission. That is the virus being present in the mouth as well as the nasal pharyngeal area. Now, if we were then to apply this approach to dengue, one could then see that since we're not destroying or preventing mosquitoes, what would happening is people would be wearing hazmats. So instead of taking an aggressive approach and following this COVID-19 approach, this is how we would be approaching dengue. So I'd like to point out then the importance of intervening early in the infection process. So that's why the key here is to stop multiplication early. and because the generation of the variants. So if we stop multiplication early, that then prevents the generation of variants. So immunization itself, and this again, what I'm saying here is not anti-vaccines, but and one of the things we learned about from these new vaccines are the vaccines are a bit leaky or non-sterilizing, because you could still be infected. You would have these breakthrough infections, and those with breakthrough infections would still be able to generate variants. So therefore, I think with this course of the talk, I think it's not limited for those who are not unvaccinated, but even the vaccinated people would still be required to use early intervention strategies. So now shift gears now to the next part, wherein I would like to look at early treatment studies, which have been shown. This is taken from this website, c19early.com. So most of the time, people have looked at early strategies. I think the current strategy, is once somebody's infected, you isolate. And hopefully I've gotten across the message that simply isolating somebody who is infected with COVID-19 is not sufficient, because that person who's isolated is continuously shedding virus particles. And somebody who's continuously shedding viral particles is shedding a lot of variants, which does not help in controlling this pandemic. So here is a table of a table of early intervention strategies. I'm not going to go through all of them. We know that, hold on, sorry. We know that there are FDA-approved drugs for it. This one's an antibody, a monoclonal antibody. Another monoclonal antibody combination from Regeneron, Ivermectin. And then we also have remdesivir, which is actually used early. It's also used in the Philippines. So these are antibodies which prevent the binding of the virus from infecting. And this remdesivir is an agent which prevents the replication of the viral particles. But I will be discussing today, without going through all detail, would be one here, Povidone iodine. So you'll see that it has high 81% better improvement even to the FDA-approved ones. And the more controversial one, of course, is Ivermectin, so which is pretty significant. And hopefully I could dispel certain myths as well, which has been floating around, and the importance of intervening early in the viral replication and multiplication processes. Hold on. All right, so now before I shift, I'd like to point out here another thing about viruses. You probably heard a lot about the CT value. If you have a low CT value, that means you have a high viral load. Early in the pandemic, we were looking at our CT values of above 33. But what is more important is actually what you call this PFU, or the particle forming units, plaque forming units, which is a measure of actual viral infectivity. So when you have a PFU of 100,000 per mil, that means actually 100,000 infectious viral particles. And that CT equivalent is that about 16. Because that early on, we were looking at CT value set about 40, and that's almost, there's nothing. And that's what they call the dead viral particles. The standard now, WHO has moved that to 25, less than 25, so that's around 100 PFUs. So they're still infectious, but the titer is very low. So once you go at about this high titer of 100,000 infectious viruses per milliliter, that's when you could then transmit it very efficiently. Now, the CT value doesn't, although this is the current way of measuring viral load, this could also be misleading in certain ways, because as you'll see in the next slide, there's one thing which we call the particle to PFU ratio. So in the particle to PFU ratio is the number of viral particles to actually which are infectious. And for SARS-CoV-2, that's 1,000 to 1 million. So what that means is that for every 1,000 particles, only one is actually recorded as infectious. Not that 1,000 of them are 999 are defective, but that's probably more due to the efficiency of the virus forming a productive infection. Now that has implications to the previous slide here, so that CT number doesn't automatically correlate with how much viral load, but physical particles. What's more relevant here is the PFU concentration. And 100,000, as I earlier mentioned, is actually very high. And once you go anything below 100, although they're infectious particles, is manageable. So if you could, and the standard for any antiseptic reagent for cleaning, that's why you have 70% alcohol for wiping out, is a reduction of 99.99%. So anything with 100,000, you could then reduce it to 100 is a good antiviral agent. Okay. So as I pointed out, so one option then is to use povidone iodine, and povidone iodine is actually locally known in the Philippines as betadine. I think there's a brand called Alphadine. It's commonly used as a gargle for bacterial and viral infections. It's safe, and no resistance has been developed against this for both bacterial and viral infections. And the reason behind that is the mechanism of action. So what povidone iodine does is that it modifies the protein on the surface of the virus as well as the nucleic acid. So regardless of what the mutation is or what the variant is, it will be inactivated. And it has been shown in vitro that within 15 seconds, if you apply povidone iodine or betadine, you reduce itself by 10,000-fold. So as I showed in the earlier slide, you reduce the number of infectious subunits. So if you have 10,000 PFU per mil, in 15 seconds, you treat it with betadine, you essentially have 0. So interestingly, late last year, there was a study published in Bangladesh looking at 606 COVID patients. They were divided in 2 groups. One was treated with 1% betadine as a gargle, as a nasal swab drop, as well as a 1% eye drop. And the other group was just given warm water. And what they found out was the group which just had control warm water did have a significant after day 7, 70% measuring by RT-PCR positivity. As I showed in the earlier slide, even if you have an RT-PCR of 32, a CT value of 32, you could still have few viral particles present. If RT-PCR, so this is measuring positivity. If you're 0, that means infinity your CT value, there are no viral particles at all. This would then show you that the control doesn't have much effect. However, if you apply betadine as a gargle, as a nasal drop, as well as an eye drop, within 3 days, after 3 days, so these are people who started COVID positive, 89% were COVID negative, so no detectable COVID. And after day 7, 98% of the 303 patients did not have any RT-PCR. They were RT-PCR negative. What's more important is those who have been treated with povidone iodine had better survival rate, so nobody died, and fewer patients were required oxygen support. So just a simple treatment then, and that could be attributed then to the inhibition of viral replication early on. So that was published. Let's just show you in this slide. Now, there was a second study, which is a different study in Singapore. This one, instead of treating people who had COVID, this one's more like a prophylaxis. So before any infection. But they were in this study, they were comparing 5 different, or I think 6 different treatments. So this was done in Singapore. They were looking at 3,000 migrant workers. They were in a dormitory. There was a breakout in infection. So they took 3,000 migrant workers who were not infected. And then for 6 weeks, they gave them this different treatment. So roughly about 500 subjects in each group. So that one had vitamin C, daily, hydroxychloroquine. It's a bit controversial as well. People said it doesn't work. And then povidone iodine throat spray. In this case, they didn't have a nasal or an eye drop. Ivermectin was done once, and a combination of vitamin C and zinc. And the results are here. The best one was the throat spray with povidone iodine, where 46% were only 46% infected, and 54% were 54% were not infected. So then the others, then hydroxychloroquine was next one. Of course, ivermectin at 64%, but I'd like to point out this was only used for 1 dose, 12 milligrams for the course of 42 days or 6 weeks. So it is also possible. It's worthwhile to note that this all these treatments here are not contraindicated with each other. So the possibility then of using this as a combination for one treatment is worth exploring. All right, so now here's the more controversial one. I think ivermectin, which has pretty much divided everyone. So as full disclosure, I'm fully vaccinated, 2 of AstraZeneca. So, but course of for the sake of discussion, I'm not anti-vax. So I hopefully this part will be treated objectively as possible. All right. So ivermectin, everybody knows, I think it's been shown, essentially won the Nobel Prize in 2015. And they're essentially composed of 2 molecules, large molecules derived from, it's an ivermectin, the class of molecules derived from Streptomyces. And it's 80% of this B1A and 20% of the B1B. And the only difference between these 2 molecules is that this just has an extra carbon. So molecularly, this is what you call relatively big. And it was discovered as an anti-worming agent. That's why it's called a dewormer. And the way it works is that it binds into, it inserts itself into the helices of glutamate chloride channels. And glutamate chloride channels are present across the body of worms. And what it does is ivermectin keeps the channels open. By keeping the channels open, there's a free flow of ions. And essentially, what happens is the worms burst, literally burst. So and one of the toxic effects of ivermectin is if somebody has too many worms, you have to regulate, or you cannot use ivermectin, or you regulate the amount of ivermectin used, because when all of the worms burst, that's what causes toxicity. It has also been shown that it's toxic if it crosses the blood-brain barrier. However, because this is a relatively big molecule, humans have a P-glycoprotein, and that prevents ivermectin from crossing the blood-brain barrier. However, there have been reports of neurotoxicity because of certain mutations in the transport mechanism, which leads to the transport of ivermectin, which causes toxicity. However, this drug has been prescribed over close to 4 billion prescriptions. In spite of that, has been very low toxicity or reported deaths. And so, as a matter of fact, I think the past 20 years, there were only been 4 reported deaths. So in terms of safety profiles, even safer than aspirin or paracetamol. So as an antiviral, so this was discovered last year and started as a controversy. It was discovered by a group here in Australia by Kylie Wagstaff at Monash University. And essentially, Kylie Wagstaff's group has been working with ivermectin and just stumbled on it as they were working with different RNA viruses as well as DNA viruses. And they found out that actually ivermectin works by blocking the importin pathway. So it binds to a complex protein called the importin, which contains 2 subunits, importin alpha and importin beta, and it prevents its assembly. And what does the importin pathway do? Essentially, it delivers protein, so it acts like a shuttle bus between the cytoplasm and into the nucleus. So it brings some proteins which are required to function into the nucleus. It seems that actually for dengue virus, it uses the same pathway to go to the nucleus and control the host cell so that it prevents the host from giving a better viral response. So that allows the dengue to take over the immune system of the cell. So they found out that ivermectin could actually block that transport and prevent dengue infection. As a matter of fact, there are clinical trials ongoing with ivermectin against dengue. So what Kylie Wagstaff did, because at the height of COVID pandemic last year, they did an in vitro test working with vero cells or green monkey kidney cells, and they showed significant reduction after 48 hours. Now, this create a lot of fuzz, mainly because the dose used for this in vitro studies were about 100-fold times the regular dose used for parasitic infections. Fortunately, a lot of doctors did not read the memo about the concentration used for these in vitro studies. So there was initial skepticism whether this worked, but fortunately there were a group of doctors, or many doctors, who thought that this might work and worth testing, and that's what they did. And there were several clinical trials done. And just like to point out, because this is a myth which has been floating around, and I'll comment on it later, is the dosage used is not the same dose used in the in vitro study. So it's not 100-fold times. In spite of that, you'll have a preponderance of different studies here. You have 63 studies. I think there are about 44 published, which show efficacy, and there's also about 5 which did not show efficacy. But overall, the treatment, especially in the early phase, has been very high. And I'm going to touch, I think, certain controversies and confusions. So I'm going through this process again, as pointed out. So you have the replication cycle here. I'd like to point out also that the key molecule here involved in the inflammatory process, NF-kappa B, which I'll show later. So it's been shown that ivermectin actually inhibits the binding of the spike protein to the receptor, ACE2 receptor. It also inhibits the binding to the co-receptor. It has also been shown that it actually also inhibits the protease involved in the replication or the processing in the early stage of replication. As I pointed out from Kylie Wagstaff's group, it inhibits NF-kappa B. It inhibits the transport of viral proteins into the nucleus to prevent control of the antiviral response. And it also inhibits the replicase. So what you could see here then is there are different multiple points in the replication process itself. I think there are people concerned, oh, if you use ivermectin, they'll develop resistance. Now, the problem with that is, especially for the important pathway, mutations would only occur in the viral protein, which the virus could control. So mutations, the possibility of having resistance against ivermectin is very unlikely, especially it's acting against different multiple points. And the likelihood of having a mutation, a resistant mutation of ivermectin affecting the import transport is very unlikely because the important pathway is a host gene and not the viral gene. More importantly, what's interesting is ivermectin, because NF-kappa B is a host protein, which acts as the master regulator for inflammation. So if the cell senses inflammation, what happens is NF-kappa B enters via the important pathway into the nucleus and turns on the genes for interleukin-6 and other cytokines. And those are the ones which generates the cytokine storm. Because NF-kappa B uses this important pathway, ivermectin actually blocks this particular phase. And then therefore that explains the anti-inflammatory response of ivermectin. And that has implications, especially in terms of cost. As people know, ivermectin is a cheap drug. And one of the things, especially in the Philippines, from what I read in the newspaper, there's a shortage of tocilizumab. So tocilizumab is an antibody against the interleukin-6 receptor. So that prevents the binding of IL-6 to the interleukin-6 receptor, which triggers a cytokine storm. However, I'd like to point out that interleukin-6 is not the only cytokine involved in the cytokine storm, although that seems to be predominantly or the dominant one. But there are anti-interleukin-1 beta and as well as TNF-alpha. So just relying on a single, very expensive drug is which partially inhibits the cytokine pathway or cytokine storm is worth noting. And whereas a cheaper drug such as ivermectin could prevent that at an earlier stage or upstream of that particular process. How am I going? All right. Okay, so that shows here. So as I pointed out, it's obvious then by intervening early in the viremic phase, we could then prevent the progression of the disease towards the severe phase. And so far, I think most of the antivirals haven't been used are the expensive ones. So you look at remdesivir, it's all still used, as well as the monoclonal antibodies, and those are very expensive, the tens of thousands of dollars. And there are limited efforts, or at least things which are currently approved or promoted. And at least I pointed out two. ivermectin as well as povidone, iodine, which could then be used to regulate this particular process, not only to prevent the progression of the disease, but also to prevent the generation of variants. Okay. Now, this is one thing I'd like to cross what earlier mentioned, the Rubicon inflammation as well as the cytokine storm. So it was proposed earlier last year by Panigrai et al. That if you look at inflammation in the alveoli, what happens is, especially with people who have comorbidities like diabetes, obesity, or other autoimmune diseases, usually they have low levels of these EETs or SPMs. So that's epoxy trianoic acids, a specialized pro-resolving mediators. Just like to point out, actually, these are intermediates of or are derived from omega-3 fatty acids. Omega-3 fatty acids are those fatty acids which you find from oily fish, such as sardines, and it is known that if you have high omega-3 levels, it is known that people with comorbidities have low omega-3 levels, which explain their low levels of these EETs and SPMs. So what happens is if you have low EETs and SPMs, the trigger for producing a cytokine storm is much higher. And once that happens, you then create this cytokine storm leading to your hyaline membrane, which is pathologically a sign of a COVID disease. What's important then is of course, it's important to have an inflammatory response to get rid of the virus, but it's important to then have inflammation resolution, and that could be achieved by having an increase of these EETs or by having higher omega-3 levels. And that has been confirmed, or at least supported by recent study showing that in a pilot study that people who had a higher omega-3 index or have high EPA and DHA fatty acids in their red blood cells, they have a lower chance of dying. And that's consistent with this, because it's also known that people like obese people who have low omega-3 levels have low immune responses. And it also then leads to they have a tendency to develop more inflammatory diseases. Okay, what's interesting though, I'd like to note is it has been shown that ivermectin, although it was in mice, actually, if you treat mice with ivermectin prior to immunization, especially if the antigen is T-cell dependent, which requires the use of T-cells, which is the case of COVID-19, these mice actually generate an immune response better if given ivermectin. So it's tempting to speculate that ivermectin might actually be complementary to immunization program. And it's also been shown, I think, recently also with respect to as a complement for immune checkpoint blockade for treatment of breast cancer. So the take-home message here as well is one preventative as one could also ensure as a layer, an extra precaution is the increased intake of omega-3 fatty acids, so that if a person has IEETs, that makes a more efficient immune response as well as to prevent the propagation of the virus, as well as leading towards a cytokine storm and complications arising from it. So this just summarizes, I think, what I've presented with regards to the stages of COVID infection, as well as the role of ivermectin. One could conclude then, essentially, pretty much, ivermectin operates on all phases. Highlighted in red here, or at least, in red, are studies based on modeling. So it hasn't really been shown that ivermectin has indeed, in the test tube, binds to the S protein TMRS2 or replicase or protease, but based mainly on modeling studies. But there are supporting studies for other aspects of ivermectin against COVID-19 and the related aspects of the disease. So now I'd like to shift now here addressing issues on ivermectin. I think one of the people or skeptics for it, which is natural in science, that claim that there are not enough randomized clinical trials and, of course, presence of bias. I refer people to this slide. I think it's a review from Bryant et al., or a meta-analysis from Bryant et al., where they used 21 randomized clinical trials, including those which show negative results of ivermectin. And more recently, I think a few weeks ago, there was also a claim. There were concerns of a particular study which showed the efficacy of ivermectin, the validity of the results, the El-Ghazar study, which was originally used in this analysis. So at least three meta-analysis which used that study re-evaluated their studies, taking out that particular study, and their conclusion hasn't changed, that ivermectin does indeed show efficacy. Now, there are the negative ivermectin clinical trial results, which is natural, but I think, and this is, I think, where it gets overblown. What you need to read it in the context of what the study usually shows. Oftentimes, what the study, and even in the paper itself, when it shows it doesn't work, they would admit that it's underpowered. What that means is there were not enough subjects to show statistical significance, because oftentimes that's what the studies show, were published, that there's no difference between the placebo and ivermectin. Oftentimes, if there's no difference, it's often attributed to the limited number of subjects used. Other limitations as well is it's below the therapeutic dose. The dose used in those studies were very low and not the dose which would give you an effect, which is then obvious. Next thing is also subject profiles. Most of the studies use subjects who have a less chance of going to the hospital. So if you're young, have no comorbidities. So if you develop COVID, the likelihood, even if you're in placebo, likelihood of you going to the hospital or developing complications is low. So of course, if you have limited number of subjects and if you're in that profile, you're not going to get any benefit, then the result will be most likely negative. The other thing is the baseline and clinical endpoints. Oftentimes, some of the studies which showed negative results, it wasn't clear whether the subjects who were in the baseline were already given ivermectin, because they were only given a clearance of, let's say, 1 week if you didn't take ivermectin for 1 week. But there are studies showing that ivermectin actually stays longer than that. So your baseline becomes contaminated, and the clinical endpoints are also different use. It's either survival or RT-PCR, or the presence of the virus. The other issue, of course, raised as well, and often keeps on being repeated, is that it's used 100 times more than allowed in human. Well, actually, that was done for the in vitro study, which was about 100-fold time. But there are recent studies which still yet to be published, when the studies were repeated with human alveolar cells instead of the human viro cells. The IC50, the inhibitory concentration, is in the range of what is being prescribed. And there was a recent study as well, systematized by a French professor, looking all studies with ivermectin, and looked at the dosing, that the current dose used, which has been shown to be safe, is still within the limits of being safe. And then, of course, the one we heard about a few weeks ago, that ivermectin are only for animals. They're not a horse. So this, everybody took this horse hook, line, and sinker. And then you see even the Philippine newspapers that, oh, you can't use ivermectin. These are for animals. Of course, there are prescriptions for horses, and of course the dose for horses, because horses are like 1,000 or pounds or kilos. And of course, that is going to be potentially toxic. But there are ivermectin formulation for humans. They exist, and they've been used to eradicate river blindness. So my recommendation, I think, and there's a lot of this good reading for me that I'd recommend, is this particular ones, the response of the FLCCC, as well as the British ivermectin recommendation group, the BIRD, and this provides really good rebuttals for those. So I think as part of science, one of the things you do is you offer this as a rebuttal, and then I wait. And I've had many instances where you present this as the rebuttal, but I've never still wait for a rebuttal to this particular rebuttal. Oops, sorry. Can you still see my slide? Hello? Oh, yeah, here.
Yes, sir. Yes, sir.
Okay, yeah. Okay, now, so going for practical purposes now. So how does it work in the real world? So here's a case of Tale of Two Cities. You probably heard it. Uttar Pradesh, population of 230 million. This is as of August 21, was preparing the slide, 6.7% fully vaccinated. And on 21st of August, among 200,000 tested, they had 24 positive cases. You probably heard early in the year, about 3 months ago, they had a peak. India was running out of oxygen supply, and what this particular state, Uttar Pradesh, had lots of deaths. But then they included ivermectin in the protocol, and then you could see here the reduction. This is the number of deaths, and this is the number of cases. Significant reduction, so that the point at 24 positive cases in 21 on the 21st of August. The other is, of course, Philippines. So we had 110 million. We're 12.7% vaccinated, almost twice than Uttar Pradesh. Again, this is the slide I got from the newspaper about almost a month ago. This is January, just to show you, to make it, to normalize it with this particular area. And you see, since the pandemic, the Philippines has never really even reached baseline. We've always been hovering. The lowest we got is probably 1500, 2000 a day. And then this is peak. Now, this is the only till 18,000. And I think this is 20,000 now, right? We're above here. But I think a week ago, we had, I think I read something that the Philippines is now allowing doctors to prescribe. So we'll see whether this would then lead to a change in. So one of the things Uttar Pradesh was doing as well was the rapid antigen testing and early treatment. So they would test immediately, and even just symptoms, they would give them this medicine kit. And the medicine kit actually contains vitamin C, D, zinc, as well as ivermectin. So that was distributed, and that was part of this effort. So another case, another tale, hold on, of two cities. Malaysia, they had a hard lockdown, and this is it. This is a Google mobility, and that has been growing. Again, this is up to September 1st. Another one's Indonesia. Of course, they've been having problems, similar thing. Again, they didn't really have a lockdown, but they introduced ivermectin earlier, and then you see that trend. Now, okay, correlation is not causation, but that's pretty strong demonstration of it. So finally, summarize. So I think what I'd like to get across, so the current mitigation strategies do not effectively address viral multiplication at speed. I would like to point out, I'm not anti-masking or anti-social distancing, but the thing is, as hopefully showed in the slides, they're not sufficient because they do not affect early viral replication and spread, and that leads to variants. So current vaccines, as we know, again, I'm not anti-vax, seem to be non-sterilizing because we have these breakthrough infections. They're effective in preventing death, but it still does not prevent transmission and viral multiplication. Therefore, even among the vaccinated, it is still important that we take early stage intervention strategies. So rather than looking at vaccination itself as a means and the one be all of it, therefore it still requires some early intervention strategies, such as possibly use of ivermectin or even povidone iodine. As I pointed out here, that early intervention strategies are complementary and may have some potential synergy. And finally, I'd like to point out here that the test and treat strategy seems to be an effective way of doing it. Currently, or even before, what we often do, especially with the Philippines, which has low testing capability, oftentimes the RT-PCR, you get the results, is delayed, and then contact tracing is not efficient. But you could then have a program where you could efficiently try to stop replication early by addressing, rather than just isolating people or quarantining them and doing the social distancing and this masking, you have to have a more aggressive approach. So finally, I think this is my last point. One of the things I noticed at the beginning of this pandemic, there was all this thing that we're in this together. With the vaccines, with ivermectin, and somehow we end up polarizing each other. Either you're called an anti-vax, you're ostracized for not or believing you're an anti-science, or you're anti-vaccination. But at the end, it seems that there might be some wisdom, which I took from a quote from Jim Collins from a book, From Good to Great. And I put it here, that builders of greatness reject the tyranny of the or and embrace the genius of the end. So rather than going into camps or choosing whether it's an or, there might be some wisdom in actually in the genius of the end, that we might use complementary and multiple strategies in combating this pandemic. So thank you for your kind attention, and I'll be glad to answer any questions.
Okay, thank you very much, Dr. Ilag, for that very enlightening presentation.
Okay,
We
thank
will
you.
proceed now to our Q&A. To those who would like to key in their questions, please use the chat box below. I guess marami din po tayong mga katanungan regarding the Ivermectin or the Povidone iodine. Maybe we can key in those questions here in our chat box. I actually have a question, Dr. Ilan.
You could call me Vic. Yeah, Vic lang.
Dr. Vic, okay po. Is Povidone iodine relatively safe to use?
Relatively safe. Okay, yeah. That's a good question. Yeah.
Okay.
In general, it's safe, but I think if you have thyroid problems, have a weak kidney issues, that might not be a good.
Okay.
So something, yeah. Sorry, yeah, it's a good point. I should have highlighted that. So that's a concern. But in general, amongst other things, it is overall generally safe.
Okay, sir. Another question. Is it still effective to those patients who are experiencing severe infections already, sir? Or can we still use it as an intervention
for those in ICU already?
Yes, or are already using oxygen or has already.
Yes, yeah, good question. So I think if you look at the slide, it's remember it's biphasic. By the time you're in oxygen, you're in that inflammatory phase. I don't think controlling the virus is the problem anymore, because you've crossed that point of no return, which is called the Rubicon. Point of no return. So you treat it differently. You treat it as an inflammatory disease, so not as a viremia.
Okay.
So early on, so I think that's why I find it strange. I think we were kind of promoting this early on. So anybody who tests positive, why not give immediately, as demonstrated at clinical study, Povidone iodine? Pag nakaramdaman lang na makate or may lagnat na konti, I mean, why not actively give it? So the point I'm trying to say is simply isolating somebody and masking is not sufficient. So hopefully I could get that across. It's important that we aggressively confront the virus and reduce its ability to multiply.
Okay, sir.
I think
to our participants, there is a question. This is from Don Pangilinan. How about the use of hydrogen peroxide? Is it the same as Povidone iodine?
Yeah, I think based from studies, there's really no hydrogen. I think there were studies in the 70s that hydrogen peroxide doesn't really work that well.
Okay.
I haven't seen studies against Povidone against COVID-19, SARS-CoV-2, but there are many in vitro as well in vivo studies of Povidone iodine, and it works within 15 seconds. I think there was a paper in the 70s on hydrogen peroxide, and it took about 1 minute before it could inactivate the virus. And again, the criteria usually, and I think it's in the Philippines as well, I think there are a lot of preponderance now of these nasal sprays. So the key to look at is to make sure that inactivation is about 10,000 fold, not 1,000 or 1,000 to 10,000 fold.
Okay. Okay, sir. We have another question po from Adenico Soriaga from ADMU. Is there a possibility that the human mutated virus is more mutations rather than natural virus?
I'm not exactly sure. You mean more dangerous than the natural virus?
I guess.
Or more mutation? But technically, the human mutated virus is more mutation. So with it's more mutations rather than natural virus. I'm not.
He means, sir, I guess what he means, sir, is will mutation be higher in humans than in natural setting? I guess that's what, because
Well,
viruses mutate.
yeah, okay, yeah. So the virus will only generate mutations when it's replicating.
Mhm.
So if it's in the air, in the environment, since it's not replicating, then it's not changing. So it's just in hibernation, so to speak.
Mhm.
Yeah, so you only generate mutations once you're replicating. So the more there's replication, the virus will make errors during replication, and that happens only inside the cell. Does that make sense?
Yes, sir.
Hopefully, I answered the question of Adineko. Oh yeah, looks like looks like it answered it.
Okay, we have another question from Hasmin Chogsayan. What's the usual dose and frequency of ivermectin as prophylaxis in the context of Delta variant?
Ooh, so I'm not a medical doctor. I could just only so the one you could go to FLCC, the FLCCC, the frontline consortium or something of critical care. And I think they upped the dose to 0.4 mg, 4.4 to 0.6 mgs per kilogram for 5 days. Oh, is that prophylaxis, you said? What was the question on
Yes,
prophylaxis?
it's what's the usual dose and frequency of ivermectin as prophylaxis in the context of Delta variant?
So I'm not. But you might check the FLCCC website. I think they have that in their eye mask protocol.
Okay, sir. Okay, we have another question. Okay, we have another question from Mr. Christopher Silverio. What can you say about ivermectin can cause liver damage? Ivermectin, I guess.
I think they did the study on that French professor. I think I forgot what the name of the professor is. And it's not associated with liver damage based on the different toxicology studies, which has been shown.
Okay po to. Another question po from Maria Therese Alonzo Dimamay. If there are more evidence of the positive effects rather than negative of ivermectin as COVID-19 treatment, how come it is still not recommended by WHO?
So I cannot speak for the WHO, but you could ask them. But from a scientific point of view, I'm on the side that I think there's enough evidence that you could go through it. But here's, I think, a good argument from this Professor Colleen Aldous from South Africa. And I think there was, let's look at, there were two studies good for comparison. That's the Bryant et al., the meta-analysis, which showed that it works. There's a meta-analysis done by Roman et al., says that it doesn't work. But when you read their relative risk calculation, one had 0.38, the other had 0.37. So almost identical. The only difference was the Roman et al., which showed that it does not work, is the confidence interval was so wide that it reaches to the point above 1.0. So that's why there's this question whether it does work. So taking that in context, so if the number is the same, the analogy I could compare it with is you have a glass and it contains 50% water. Okay, so one looks at it, it's 50% empty. The other one looks at it as 50% full.
Oh.
So that's where we are right now, in my opinion. Now, what Professor Aldous, Colleen Aldous said that, okay, so in statistics, there are two possible errors. One, you have the type 1 error, which is false positive. So in the case of a false positive, you think ivermectin works, but actually doesn't work. Okay, so what's the cost then of a type 1 error? One, it's probably too expensive. It's probably would cause deaths, or it would cause some other things. So if I make a type 1 error, what would be the costs? Death, too expensive, or some other thing. Okay, now the type 2 error is the false negative. You're not using it, but actually it works. So this is like the sin of omission type error. So the question then is, by not using it, it would cost you something. Does that cost you? Would somebody die if you don't use it? Is it too expensive if you don't use it? So based on those two assumptions, two choices, what would you choose? So I don't have to explain that to you, but I defer that to how you would. So that's a one way of analyzing it. So the question then, does ivermectin, if you don't use it, if you use it, if it doesn't work, how much will it cost you? And to save life. Now the cost then, if it type 2 error, and if ivermectin does work, but you're not using it, that means people are dying, and people, because it's cheap, the alternative is more expensive, you're losing more money. So that'd be the simple argument. So I leave it to you guys how to decide that. But of course, I have my own way of my decision or choice.
Related to that then, sir, I have a question then po. Were there studies also done here in the country involving povidone, iodine, and ivermectin here in our country using human subjects or animals?
Actually, I mean, very good question. We tried to propose that last year. But we couldn't really get a takeoff. I mean, with povidone iodine. Really, I mean, we tried to do it, but there was no uptake.
Maybe we can include, sir, that
Yeah,
study,
and
sir.
I think there are plans on the ivermectin. I think it's ongoing. I think DOST. So I haven't seen the protocol, but I think again you have to put in context the limitations I pointed out early on, why things turn out negative. You have to make sure that the dose you use is the right dose, and I think there's also tricks. I think one of the things they want to make sure when you take ivermectin, you have to take it with a meal, because it increases bioavailability by 2.5 times. The reason it's not taken with a meal when you're treating a parasitic infection, the worms, is because of the concern that if there's a lot of ivermectin and you have a lot of worms, it would potentially be toxic.
Okay, sir. We have another question here from Davy Ann Calderon. How can we stop the production of new variants of the virus?
Yeah, so as I pointed out, if somebody already tests positive or have symptoms, if you treat them early, you prevent the replication. I think I had that slide which showed that the longer you let the virus replicate, the more errors the virus will make, and that's how it generates mutations. So just to point out, so vaccination or drugs do not cause mutations. It selects for the mutations, but the mutations come from allowing the virus to replicate. So the key message is, if we could stop the virus from replicating early by intervening early, then we could prevent those generation of those variants. So the bonus as well is, by treating early, you prevent people from leading to progression of disease. You don't enter that second phase. And in the context also, I think Athena, it's also been shown that natural immunity is more durable than vaccination. So natural immunity, if you get infected, the immunity you get is actually better than vaccination. Now, hopefully people don't misconstrue what I'm saying. So I'm not promoting that everybody goes out there and be infected.
Get COVID.
Yeah, but this has implications, I think, especially with the Philippines. First, we have limited vaccines. I think our testing is a bit slow, but we don't even know how many already. I could tell you that there are a lot of asymptomatics, and those asymptomatics already have good, robust immune system, immune responses already. So technically, you don't want to waste your vaccine against somebody who's been infected already, because that person already has a good immune response if he or she gets a breakthrough infection. So you could then save the vaccine for somebody else. Now, at the same time, if we go treat early, anybody who gets infected already technically is, quote unquote, naturally vaccinated. But one way you could do, by treating early, we prevent them from going to the hospital. So we hit two birds or several birds with one stone. by treating early. So again, I'm not promoting that people should not get vaccinated. I mean, there are other concerns, but there are different strategies we could use. We just cannot rely on, oh, everybody should go vaccinated.
Okay.
So,
Okay.
yeah. So hopefully.
Sorry. Go ahead, sir.
Hindi, okay lang. Yeah.
Okay.
That's it.
Shall we proceed to the next question, sir?
Yeah.
Yeah. Okay. We have a question here from Hasmin Chogsayan. Why is it that COVID is so dynamic as compared to other viral diseases? H1N1 mutation takes years before mutation, but COVID just mutate drastically.
Okay. So H1. So there are two different things. I think not exactly. So influenza, H1N1. So this is influenza, I presume, right? H1N1.
Yes, sir.
Is this H1N1?
H1N1 influenza, yes.
So actually, H1N1 is not a mutation per se. So H stands for hemagglutinin and N is neuraminidase. So the flu virus is different than corona, than SARS-CoV-2. So SARS-CoV-2 has only one genome. Influenza is a bit different beast. Actually, influenza has 8 different segments of RNA. Okay, so when in fact it has 8 different segments. One of the segment contains the hemagglutinin. The other one contains the neuraminidase. So what happens with influenza, it actually packages 8 different genomes in a viral particle. So the error rate, I think, as well for influenza is not as high as with this, with the coronaviruses. And in terms, I think, if I'm not mistaken, the efficiency of infection is not as great as this, as COVID-19 or SARS-CoV-2.
Okay. Another question po here in Zoom from Christine Eden Sevilla from FNRI. Is it correct to say that if more people get vaccinated, virus circulation will decrease and thus may lead to fewer mutations in the future?
Let's say more people get vaccinated. Okay, so this is very good. So this, I think I'm not exactly sure, but this is what we know about this. The vaccines right now do not prevent transmission. And it seems that it does not also, so the fact that we get viral breakthrough infections, it definitely is useful that it prevents death, because somebody who has been immunized is already some pre-existing antibodies. So I don't think you could, and the reason behind that is it seems that the T cell response of these vaccines are not very good. The T cell response will make, if it were good, will prevent replication actually. Because when a cell is infected, before it's even releasing viral particles, it'll already be destroyed within. So actually there's a concern, and I think there's a certain group which is valid, because based on Marek's disease, which has been observed at chicken, that if you vaccinate somebody with a leaky vaccine, you enrich for variants. And this could be the case that if you have the antibodies are against one particular wild type, if they are selected, there would be variants. If you allow them to replicate, there will be more variants, and that there will be then selective pressure against the wild type, and the variants would then be allowed to replicate or propagate themselves.
Okay,
Yes.
sir. Okay. We have another question po from Sonia De Jesus. So talking about the efficiency of ivermectin, how about its contraindications?
I'm not sure exactly what the contraindication is. I think anything which would make your blood-brain barrier permeable would be a contraindication. I think blood thinner, I heard, might be a contraindication for it. But overall, I think it's been safe. But again, I'd like to point out, so I'm not making medical recommendations. I think it's better to talk with your doctor.
Okay, sir. So we have another question po from Carla Jasmine. A follow-up to the current topic, is the immunity enough for the Delta variant now?
The immunity from the vaccine
Is the
or something?
immunity enough?
You mean
Sir,
from the
from
vaccine?
the natural immunity when
Nat-
you already had the COVID itself.
Yeah, I think so. The advantage of the natural immunity is, I think, again, this is my speculation, that's why it's more durable. I think the T cell response is better. Because when you get infected by the whole virus, you generate T cell epitopes against all proteins. And it seems that the nucleocapsid, which is found inside the virus, is a better epitope for generating T cell immune responses. Note that the current vaccines which we are using, the mRNA and the adenoviral, AstraZeneca, is only the spike protein. Of course, there's Sinovac and I think Sinopharm and Bharat, but I know the performance hasn't been that satisfactory, even though theoretically they should be the better vaccine because they include the intact whole virus. So the only explanation I could think there is, I think the adjuvant, the thing which you mix with the vaccine they use, is not a good adjuvant, which could explain why you have a poor T cell immune response. But for natural infection, I think from the Cleveland Clinic study of 52,000, I think it's good. It's good as even a year, I think, if I'm not mistaken. More recent studies as well, so it's more durable. Then, of course, because you have B cells, your memory B cells and memory T cells, so that gives you, I think, that additional power for those who have natural immunity.
Okay, sir. We have a question here from Antea Jane Kaha. Is it true that the virus only lasts in your body for 14 to 21 days? That's why you will be quarantined within the said days if you're infected.
Yeah, exactly. So this is where I have a problem, actually. So it has been shown that other people, infected people, shed virus even after 53 days. So that's why I'm a bit queasy about this thing that once you test positive, the only thing you do, quarantine, social distance, and stay home within 10 days you're infected. So, and then you assume, there's always this assumption that after 14 days, you're definitely cleared. I think in general, overall, that might be the case. But I think for this particular virus, as well as it's the way it's mutating, it's better to intervene early. So once you test positive, I think why not just give an early intervention, whether it's Povidone iodine or Ivermectin. People don't like it, but there are other things I think which could be used.
Okay, so for our last question, sorry, we are on our last question now due to time constraints. This is a question from Fidel Malbas. Doc Vic, you mentioned earlier in stages of COVID-19 that omega fatty acids source in fish like tuna and salmon. This means that we have to eat fish instead of meat in this COVID-19 pandemic. Due to your study, people affected with COVID-19 have comorbidities of diabetes, hypertension, etc. Maybe they are not fond of eating veggies and fish instead of eating meat all the time.
No, actually, it's a very good point, I think Fidel made. So I think that's one of the things I think which has to be promoted. And actually, you don't have to eat tuna and salmon. Actually, you know the one which has the highest sources of omega-3 is actually sardines. So sardinas. Actually, so you don't have to buy this fish oil. So there's sardinas. Has a high amount of omega-3. So it has been shown. So actually for diabetes, so if you have an omega-3 index, a measure of your EPA and DHA of red blood cells, if you have an omega-3 index about 8 and above, actually decreases your chance of a heart attack. It's also been known high omega-3 leads to lower hypertension. It has lower blood pressure. So yeah, you're right. So Fidel's right. So actually what has to be promoted is actually increased intake of omega-3 as well as vitamin D. Again, part of the thing, if you have this high omega-3, high vitamin D, once you get infected, you'll be almost having a natural immunization. So you should not panic when you have, not to be fearful, but by taking this extra precaution. So I don't think it's a negative. Once you get positive, it's not like a death sentence unless you have comorbidity. So that's why the incentive is actually have to watch out your health, and there are preventive ways of doing that and do it early.
Okay, so vitamin D and omega 3.
Omega 3, yeah.
Okay,
That's known,
yes.
actually, yeah.
Yes, sir. Okay, so I will now proceed to our to present the certificate of appreciation to our dearest speaker. May I call on again Dr. Annabel B. Briones, our ITDI Director. Dr. Annabel?
Can you please flash on the screen the certificate so Doc B can...
Okay, ma'am. Just for a while. Hold on po, ma'am. I'm sorry I was not able to prepare it earlier.
Ano yun? Yung?
Yung certificate, sir. Okay. Can I flash?
Yeah.
Ask Joven.
Okay. Okay, ma'am. Can you see it now, ma'am?
Okay.
Yep.
Okay. Okay.
May I award the certificate of appreciation to our guest, our speaker, resource speaker for this afternoon's VIP webinar series. So Republic of the Philippines, Department of Science and Technology, Industrial Technology Development Institute presents this certificate of appreciation to Dr. Diodovico L. Ilag, Mulawin Creek Ventures Limited, Australia, for sharing his expertise as resource person for the webinar entitled Early Interventions Against COVID-19 as part of the establishment of the Virology and Vaccine Institute of the Philippines VIP program of the Department of Science and Technology, held online on September 16, 2021 via Zoom, given this 16th day of September 2021. Yours truly, Director of ITDI. Thank you very much, Dr.
Okay, thank you.
B.
Thank you, then. Thank you, Dr. Annabelle.
Very informative webinar. And we have a lively discussion.
Thank you so much po.
Thank you, then, Tina.
Thank you so much, Dr. Briones. And again, thank you, Dr. Ilag, for your very informative and interesting talk on the early interventions against COVID-19. I hope all our participants was able to learn, take home some valuable lessons with them from this webinar.
Okay,
Now,
thank
to
you.
formally end our webinar this afternoon, may I call on Engineer Reynaldo L. Esguerra, the Chief of the Environment and Biotechnology Division, for the closing remarks. Sir Ray?
Good afternoon, Doc Vic.
Thank you.
Kung hinanap yung
Afternoon.
yung video mo, ayun. Nice to see you again.
Salamat din.
Okay, so, well, pag-ilan na ako magte-thank you to you for during this webinar series. So, again, maraming salamat for imparting to us I would say a fresh outlook on some of the alternatives that
Okay.
look into. Kasi sabi nga, sa science, we have to look at all sides, hindi lang sa isang side. So maybe our participants could look at the references that you mentioned and really digest. Of course, yung we did not spend 2 hours ngayon, pero siguro going for them to go through what you've shared. Pero ang importante po na thank you natin kay Doc Vic is for him being part of the Balik Scientist program. He's one of the 7 scientists, and this is the first time that he has spoken sa ating webinar series. The other 6 had spoken, and he's the only one from down under. Siya lang po yung, siya lang yung Australian, galing sa Australia. The other balik scientists natin are either from UK or the United States. So, again, maraming salamat, Doc Vic. And then,
Wala problema.
sanang, when he showed about the treatment, nakita niyo lang yung vitamin D. I recall when we had one of our meetings on a Saturday, na-mention niyo nga, vitamin D. Where is the free source of vitamin D? Di ba alam naman natin lahat, nung bata pa tayo ay sa araw. Unfortunately, with the quarantines and the lockdowns, kaya sa bahay lang lahat ng tao, hindi naaarawan.
Yeah,
So,
exactly, yeah.
mukhang there has to be some thought that has to be given on people really have to still go out of their homes. And again, another important lesson that maybe we could look into is yung sabi niya, address the viral replication and spread. So ang tinatry natin is preventing it from going to us. Kaso if it's still there, nagre-replicate, nag-spread, in fact, I was trying to find, unfortunately, I'm an engineer by profession. Also, ang sa amin talagang mga measurements. So I'm trying to find out what is the particle size of the COVID virus and what is the pore size of the mask that we're using. Parang ang alam ko mas maliit pa rin yung
mas maliit ang virus. Yeah.
Yung virus kaysa doon sa pore size, no. So it's really just a deterrent, pero there's still that even if you wear mask, you could still be infected. Kaya nga yung replication and spread, addressing that is very, very important. So ngayon, I'd also like to take this opportunity naman to thank ang ating host, si Athena, and also our other colleagues at ITDI sa aming Planning and Management and Information System Division, saka sa Technological Services Division, and of course our director, who has been, not only is she our director, but she's also the program leader of this establishment of the VIP program sa Philippines. And again, also I'd like to thank si Joven. I'd like to recognize si Joven. Siya yung tagagawa ng aming mga certificates that we award to our speakers. So with that, again, siguro it's close, but please everyone, watch out for the forthcoming webinar series. Hanggang 2022 po ito, April 2022. This will run until 2022. And I'm sure Dr. Vic would also be featured at least 2 or 3 beses pa. So again, Doc Vic, I forgot to ask, from what province are you?
Laguna, Los Baños. Tubong Los Baños ako.
Kasi some people would say, dalawa yung Dr. Ilag na yan. Pero pag sinabi naman nung sinasabi ni, ay baka yung kilala nyo yung mas konti yung buhok.
Ako yung puting buhok.
Siya daw yung puting buhok. Yung isa naman ay
walang buhok.
So, again, maraming salamat po and
thank you rin.
And stay safe everyone.
Okay, salamat rin.
Good afternoon po.
Okay.
Thank you.
Thank you.
Thank you very much, Dr.
Yeah. Ano yun?
Yes, thank you.
Walang problema. So, Sabado ulit.
And
May presentation ako sa inyo Sabado, no?
Thank you.
Okay,
Thank you
Athena,
so
yung
much. Okay.
in-email ko yung kay Rika presentation kung may gusto may kopya yata or something.
Okay, sir. We will distribute it to our participants, to those who want to have a copy.
Kasi yung references na sabi ni Sir Ray.
Yeah. Okay, sir.
Baka release mo. Yeah. Okay.
Paki-email na rin din yung ibang questions na hindi natin na
Yes, ma'am.
Sagutin na lang sa email nila.
Yes, ma'am. To those who have questions to pala na hindi namin nasagot this webinar, maybe we will consolidate them and send it to Sir Ilag, to Dr. Ilag, so that they can. Yes. Okay. Okay. Thank you so much.
Okay, salamat.
Bye.
Bye-bye.
Bye-bye. Andyan si si Chris. Si Dr. Silverio.
Sa FB Live siya.
Ha?
Kala ko FB Live. Sa
Ay, FB
Facebook
sayang.
Live.
Kasi
Oo.
Okay na.
bye, Doc.
Okay, bye. Tapos na ating picture na.
Ulit, Doc.
Tumitilaok na ang manok, hudyat na ng pagpasok. Paglilingkod na walang kapalit, sa bayan ng aming hatid. Tara na, kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay magkakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Anumang hamon ay haharapin, mahirap man ay kakayanin. Sa pinagsamang lakas at galing, tagumpay ay mararating. Tara na, kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay magkakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay magkakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Ang pangarap ko ay maging piloto.
Pangarap kong magkaroon ng chief alternative for personal hygiene.
Pangarap kong magkaroon ng mabilis at murang transportasyon para sa lahat.
Pangarap kong masagot ang malnutrition. Pangarap ko pong magkaroon ng effective communication means for emergency.
Pangarap kong ma-maximize yung renewable energy source and to reduce the carbon dioxide emission.
Pangarap ko pong maging scientist.
Ngayon na o simulan na, humanda sabay-sabay akyat. Hawak kamay tayo'y angat, lilipad. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Maayos na bukas para sa Pilipinas. Maayos na bukas ating iparanas sa bayang Pilipinas. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan.
Tumitilaok na ang manok, hudyat na ng pagpasok. Paglilingkod na walang kapalit, sa bayan ang aming hatid. Tara na, kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susunod.